Hello, everyone, and thanks so much for the opportunity. Thanks for organizer. It's a pleasure to share our recent work with sorghum drought response using a systematic approach. This one actually is working. Thank you.
It's working now?
Yes, we're okay.
Good.
Thank you. Sorghum is one of the most important cereal crops in this world, and it's also important seed grass for grain growing, cellulose, biomass production. It is also one of the most best adapted cereal to the water limited conditions. So it serves a good system for studying the drought tolerance in plants. There are several high quality sorghum reference genomes being published, along with about 400 resequenced genotypes. So this also provides a genomic basis for systems biology studies. So let's now focus on a little bit about the topic about the drought. So drought, we know, is a very complex stressor that could induce various ranges of the molecular responses. So in general, that there are two major classes of genes get involved, including the regulatory proteins, such as the transcription factors, protein kinases, and they could bind or interact to the downstream genes, as known as a lot of functional proteins. Those functional proteins get involved to certain biological process, finally forming to the drought tolerance. Because there's many genes involved, many complex gene networks. So mining the genes enhancing plant performance under drought can require a lot of work, requires multiple lines of the evidence. In general, we know that the plants perceive the drought signaling under the water limited condition. The first reaction that we can actually observe is the phenotypic changes. So for instance, you can notice such as reduced yield. So from a phenotype aspect, we notice many changes. Behind those changes, there could be many reasons. And I think, as just mentioned in many talks, that we know the genomic variations could be one of the main contributions. So there are a lot of SNPs or indels variations within respective genomes. And these changes could further lead to the high or low expression of some particular genes, such as a lot of genes evolve to the certain process. The other aspect that people may don't talk that often is about the epigenetic mark. So we know we have DNA methylation, histone modification, but here we're going to show one example of the RNA modification, which are the biochemical changes of the RNA, and these changes could further influence the gene expression. So we know there's a lot of connections among these layers of information, but how we can find the genes of interest from tons of them. So I'm going to propose a little bit thing called the co-expression network approach. So this is actually kind of an algorithm to help you to divide the genes into different clusters based on their expression. So the co-expression network going to cluster genes with similar patterns into a module, and each node represents a specific gene. And this kind of lines are the connection. But remember, this is not true like the interaction, just say like connection, because they kind of express very similar. So how we can know if they're true, like interact to each other? So first, we probably gonna do some literature search based on previous studies. We find, oh, there are a lot of stress regulators from previous study, they kind of exist in this network. Also, we could notice there are some genes that are highly correlated to their traits. And there are some of the genes associated with the structure variations. We found all the things they are kind of appear in our co-expression network. So maybe we can perform some functional validations. And now you can see these are like the true interaction, but not only the mathematical connections derived from the network. And finally, we can hopefully find the genes enhancing the drought tolerance. So let's come back to the project a little bit. And as said, we focus on sorghum. And in this project, we have 6 sorghum accessions with different genetic background. We plant them, and on the 2 conditions across the 4 time points, the leaf tissues were taken for RNA extraction for RNA-seq, as well as for other phenotypic traits measurement and metabolites abundance profiling. As said, we're going to use a systematic approach. So we have this respective analysis, and more importantly, we're going to build up a regulatory network to build up the co-expression modules and identify some of the hub genes involved in this process. Okay, So the first thing, let's come back to the phenotype. The first layer information that we find our sorghum accession display the significant variation in their response to the drought. We noticed the reduced biomass for sure in most of the accessions, but more specifically, we noticed that one of the A accession here, they exhibit seriously wilted leaf tissues. Also, we noticed some big differences regards the root length and root height from the A relative to the others. So now we can see maybe this A seems like a very bad performer. So along with some relative good performer, and this accession should help us to uncover genes associated with stress resilience. We also have molecular phenotype, as just mentioned. Here we mainly focus on metabolites and some liquor-derived phenotypes. So liquor-derived phenotypes are a lot of photosynthetic related traits. We know that under stress, a lot of changes happens during the photosynthesis. Based on the PCA of the metabolites that we notice, actually the accession A kind of stands out from the later stage when they compare to rest of our accession across different conditions. Also, the chlorogenic acids and sucrose are the primary factors leading to the PCA pattern you observe here. And we know these two are kind of two common stress responsive metabolites. So based on the liquor phenotype, and we notice the water use efficiency, for example, the gas exchange data, they're kind of very different, especially at the terminal stage during the plant growth. So these traits actually indicate the genotype was severely stressed, especially at the later stage. So now having covered the first layer information for the phenotype aspect, there are for sure the great strong genotypic effects, particularly for the A accession could be a central topic. So then we move on to the transcriptome and the genomic variations. These are the two important aspects. The hierarchical clustering of the global transcripts, you can see that review the very differentiation of the transcriptome among 6 genotypes. So as observed in metabolites, we also noticed the A accession at its initial stage is very similar to the other, but the later stages, they are separated, they're clustered their own, which is kind of match the observation from metabolites. Also, we annotate the genome-wide variations, including the SNPs and indels within our 6 accessions. We identify the the wide distributions, especially for some of the unique variants from respective genotypes. Okay, then we're going to move on just to mention a little bit about the RNA modification specs. So again, RNA covalent modifications are the biochemical changes to chemical changes to the RNA, and we know that the modification could induce RNA decay and further impact the transcript abundance and the translation efficiency. So here it's pretty like the way we're doing is it's a PC approach. Why are we using the global modified transcripts? We check their modification status, and we plot this PCA. So again, that the A accession seems kind of stands out. So simultaneously, we took the same sets of the transcripts, but we actually took the transcripts abundance of this data to replot the PCA, and we identified a very similar pattern in regards to their modification status and the transcript abundance. And this suggests a very close relationship between the modification and the mRNA transcription. So far we've been covered the 4 layers of the respective information, and so how we can make a good use of them, how we can integrate everything together to get new knowledge about drought tolerance. So again, here we're going to use network approach to construct regulatory networks as a framework and integrate them separately. So here's, sorry, this is a relatively busy picture. So first we have a co-expression network, and this column represents a different module, means different cluster of the genes. You will see that some of them are possibly or negatively correlated to this trace being measured, being represented by this heat map. And also we have some genes are highly enriched in some certain modules, So for instance, here we notice there is one module called the dark magenta, and it has actually a pretty high correlation to multiple traits. Also, there are a lot of genes involved in stress response to this particular module. More importantly, when we check the global-wide expression of genes within this module, we find actually the genes in this module for accession A, they respond very differently, especially at the terminal stage compared to the rest. So this will be a very interesting start for us. So we select modules. Again, that's the groups of the similar expressed genes with multiple traits association signature for analysis. So what kind of genes we can find within this module? Here's just a quick look at the network being built up. So first we have the co-expression. Again, that's just the correlation association. We know genes co-expressed, but we don't know if they are truly interacted. But we take advantage of some of the data source from the protein-protein interaction database. We know these are some protein interaction has been confirmed before. Also, we have some transcription factor to downstream gene binding data. And these are also some confirmed gene interactions. By layer this two information on top of the co-expression, we got more idea, which help us to pick up some more interesting genes. So here's one example of interesting transcription factor called the CDF. And this one we notice actually very broad connection, interaction to genes evolved to several different types of the biological process. The overexpression of these genes has been characterized in rhabdomyosis, where it could induce the tolerance and other abiotic stresses. And this might be an important hub gene for further studies. So the last slide I would like to show is when we have focused on the CDF3 at the moment, we also check their direct targets. Here are 9 genes of examples where we find their expression are also highly correlated to the CDF3, and they are targeted based on the transcription binding data. So as you can see, in some of the accessions, they have pretty high expression, but in most of the A accessions, they are really low. And this is also corresponded to multiple phenotype data we have observed. So obviously these target genes are potential candidates for further analysis and potentially can engineer the drought tolerance in sorghum and other crops. A quick summary that we have been using omics approach or like systematic approaches, uncover the several layers of information, highlights the genotypic effects, particularly the accession A. Our integrated network approach helped us to significantly reduce the number of downstream genes into only one module. And also we harvest one important genes, the CDF3. This could be a key regulator leading to the diversified drought response. And now we're working on some functional studies, experiment. So we believe that the validation of these genes will be ideal for engineering drought-tolerant genotypes. At the very end, I'd like to acknowledge the great support from Nelson Lab as well as our collaborators. And I would like to take any questions. Thank you so much for your time.
Are there any questions?
Thanks, man. I have 2 questions. Is accession A like phylogenetically distinct from all the other accessions? And then my second question is CDF3 like differentially expressed across your different accessions, or is it pretty stable?
Yeah, these are good questions. So first, we think yes, the A genotype is for sure because we took them for a very large phylogenetic tree. So they are like actually each of the accessions they come from the different like sub branches. Yeah, but we're gonna probably also redo just a local phylogeny to see like if A is more distant to the rest of 5. And for your second question, yeah, for CDF3, because CDF3 gene is also within this module. So this module actually, the CDF3 actually has a very similar pattern as you see here, because it's identified within this module. So the expression is relatively low within at the terminal stage as well. Yeah, thanks.
Are there any other questions? So whether because your section A is so different than the rest of your five sessions, I was wondering, does that reduce the power of your analysis too? I mean, because you're using, let's say this session A is so much different. It stands out compared to the rest of five sessions, and you're based on this one distinct session. I mean, would it be better like if you have two or three session like different from the remaining?
Yeah, so I think this is yes, this is actually the point we actually while we got 6 accessions, we actually don't have a very clear idea yet, but just say now, because for sure you could learn something from the superior accession. But now because A is just kind of a worse performer, we can still learn from it. Meanwhile, we're also thinking, oh, if maybe CDE, they are relatively better, we can, as you said, we can pick another 2 as another reference. But now just say, it just does not contrast that much as the A shown here. So we just have maybe more focus on it. So also to address your concern, you said, oh, if there's might be, because you said potential power loose because there's only one. But actually, we examine from multiple layers. Like if there's just a coincidence, maybe metabolites different, but maybe other layer information, they're more consistent. But now we can see it's like from multiple layers, they exhibit the difference. So I think that's actually one of the interesting stuff that we want to look. Yeah, thanks.
So for these 6 accessions, how did you pick that up? Based on the regions or just based on the phylogenetic traits, like where they are from, or they are different in drought response and
Yeah, I think this is a very good question. So there's actually several like preliminary work have been done by our collaborators. So they first actually picked about 20 to 30 accession. So they have like a pre-trial to grow them also in the field to check their stress response. So I'll say the main reason for picking this 6 are for sure they have diverse level of the stress response and also maybe be considered about, okay, the availability of the materials, every different stuff, and they kind of narrow down to the 6. Also because, because we have the leaf tissues for phenotype, for metabolites, RNA-seq, it's actually a little bit hard, like for considering about the labor, right? It's hard to get all these samples processed for all these things. So yeah, it's like combination reasons finally getting this 6. Yeah.
Now you pick you found A is an outlier. Is that you just from the regional data? Is that really different from the rest of your collections?
So sorry, can you say again for the?
Now you found A is an outlier, just from your data. I mean, just from like it's a region, or it's different, totally different from the rest of the.
So the demographic information, I think this one is actually from Nigeria. So it's actually, I think we have 2 and 3 US native accession, and this one is from like African accession. There's other 2, I didn't remember clear for the geographic, but they have different genetic background and geographical origins, I think. Yeah. Thanks.
Are there any other questions? I have one if no one else has any. So I noticed with the intercellular CO2 data and the water use efficiency, A was kind of funky things were happening. And I was curious if any stomatal, like there's any differential expression with A or upregulation, downregulation of anything with the stomata. I don't know if you've gone that far into the data yet.
I think we did have several, when we talk about the stomata, there are several actual indicators, right? You can check like CO2, NADA assimilation. We did identify some changes, and also when we check the differential expressed genes, that there are some like genes enriched to this process. So this is sort of maybe as expected because it's severely stressed.
Yeah.
Okay.
Awesome. Thank you.
Thank you.
Awesome. Thanks, Dion.